Hello friends, I'm Yamini. Welcome back to my channel. In today's video, we are going to study about the bacterial growth curve. Here we will take E. coli as an example for studying the growth curve. We know that E. coli is a gram-negative facultative anaerobe bacteria which is rod-shaped. It is cultivated on LB medium, and we know that the bacterial population undergoes exponential growth, right? Exponential growth, it will start from a point, then will reach a higher point, and then stationary phase, and then it will decline. So this is what is known as the exponential growth. Now bacterial growth is divided into 4 phases. The first one is your lag phase, followed by the log phase, and then the stationary phase, and ending in the death phase. Okay, talking about the lag phase, it is the period which is immediately after the inoculation of the bacterial cell into fresh culture media. Okay, what happens in this process is that the bacteria adapts to the growth conditions and synthesis of RNA, enzymes and other molecules occur during this phase, and there is no apparent cell division. So this is what your lag phase is. We will see in the chart. You can see here. Here is the time on the x-axis and the log of number of bacteria on the y-axis. Then we plot it. So this particular phase in which the bacteria is preparing itself for dividing is called as lag phase. Okay? Now the second one is the exponential or the log phase. Here all the cells divide regularly by binary fission. Since we are talking about the E. coli, they are dividing by binary fission and showing the balanced growth. Again we'll tally it with the graph. See, this phase is the log phase or the exponential phase, right? Here the bacteria are dividing. The third phase is the stationary phase. In the stationary phase, the population growth is limited. Now, why this population growth limits? It is because of the 3 factors. First one, exhaustion of the available nutrients. What happens is that in the culture medium, the availability of nutrients now begin to decline. Why? Because there are more bacteria which consumes that. Okay? Now the second one is the accumulation of inhibitory metabolites. You might know that this stationary phase is the phase where we synthesize antibiotics are synthesized from this stationary phase, right? So here the accumulation of inhibitory metabolites or the ANT products take place, okay? And the third one is exhaustion of space. Right? Means the biological space is now exhausted. The reason being that because the cells are already divided and they are now more in number. So this is what is called as your stationary phase. Right? There is no extra growth seen, and what we see is that the graph becomes a straight line here. Right? And the last one is your death phase. Talking about the death phase, during this, the number of viable cells decreases, right? And the reverse of growth during the log phase take place, right? So here what happens is that decline phase starts, okay? This way. Now, so this is how a bacterial growth curve look like. 4 phases: lag phase, log phase, stationary phase, and the death phase. Generally, the antibiotics are synthesized from this phase. Okay, here is your time on the x-axis, and on y-axis we take the log of number of bacteria. So that's all for today's video. Do subscribe the channel if you like the video. Thank you.